Moving on to the third talk for this symposium, the next speaker is Professor Yanghee Wu. She's a very illustrious surgeon scientist with expertise in robotic surgery and gastric cancer. And she did her training by graduating in Princeton University with a BA in political science. Then went on to do her MD at Drexel University Medical School, did her general surgery residency at Columbia University Medical Center, and went on to do a research fellowship at Memorial Sloan Kettering Cancer Center under the tutelage of Professor Yuman Feng, and as well as a clinical fellowship at Severance Hospital, Yonsei, under Professor Woo Jin Hyung as well. She currently is an associate professor of surgery at City of Hope. She's the vice chair of international affairs, the director of the gastroenterology minimally invasive therapy program in the division of surgical oncology. And she's doing a lot of work in the realm of novel targeted agents. And today she's going to talk to us on gene therapy using oncolytic virus for cancer. And she's going to speak about the recent advances in this field. Professor Wu, please.
Thank you so much for the kind introduction. It's a pleasure to be online here with you all and to participate in the International Surgical Oncology Symposium. To talk about a topic that I only discuss in the lab usually, but we are quickly coming to clinical trials. So I wanted to share a little bit with you about the work that is going on in our lab, as well as the field as where we are today in oncolytic viruses and gene therapy. These are my disclosures. One thing I want to point out is that the virus that was discovered in the Fong lab by Dr. Chen and Dr. Fong is currently under license at Immugene. I have no financial connections to this company or this virus, so I can present without any conflict of interest. So oncolytic viruses for gene therapy have been long explored for cancer treatment, and now with emerging changes in technology has become sort of a hot topic for cancer gene therapy. It has, oncolytic viruses have inherent properties for cancer targeting. They selectively replicate in mice cancer cells and leave normal cells unaffected usually. And they utilize abnormalities in the host cancer cell genome. They're able to stimulate a robust complex immune response in the host, which both limit direct oncolysis by clearance of the virus, but also this is where the opportunity arises to enhance anti-cancer immunity. And the genetically engineered virus that we are talking about today for cancer gene therapy is done to decrease virulence but improve efficacy. It's improved quite a bit with better viral gene editing techniques, more efficient oncolytic viral production, improved understanding of the immune tumor microenvironment, and we have more targets to go after. So there is an enormous opportunity for oncolytic viruses to deliver more effective cancer gene therapy at this point. There are multiple strategies to enhance anti-cancer immunity using oncolytic viruses. As you can see on the left, presented by a review in trends in immunology, all aspects of immune stimulation, immunogenic cell death, inflammatory cytokines, the OBs can carry stimulatory genes, transgenes, and express these proteins by harnessing the host cancer cell genome. So like GM-CSF, IL-2, IL-12, enzymes that degrade the stroma, even radiolabeling agents can be put onto the cell surface of the cancer cell to be imaged. And so this is all the strategies employed by oncolytic viruses. And these are some of the viruses that we know have been identified as cancer killing. And so adenovirus we hear about all the time, and HSV, herpes and vaccinia. Genes are deleted, unwanted genes are gone, and then inserted transgenes that we want. So in 2005, the first recombinant oncolytic virus actually gained approval in China for squamous cell carcinoma, head and neck, and esophagus. This is an adenovirus with 2 deletions, and it is currently in use. I think they had planned for a long time to do a lung cancer trial. I don't know exactly what happened to that trial. But most exciting in the United States in 2015, TVEC was approved for the treatment of unresectable melanoma, showing a robust response rate and survival benefit. And so this is a genetically engineered herpes virus that is the first one to be approved and the only one oncolytic virus approved for use in the United States. It has 2 genes deleted. Usual genes for neurovirulence has been deleted, with 1 addition gene that able the virus infection causes release of GM-CSF. And right now there are numerous other OBs currently under clinical trial and many others being developed in preclinical stages, including our novel CF33 platform of sumeric orthopox viruses that I will speak of shortly. So some of the trials in immuno-oncolytic viruses, as you can see, you know, the vaccinia viruses, herpes virus, measles, adeno, actually carry now the transgenes for immune therapy or for imaging, and the functions are to activate the immune system, and stimulate the immune system. And they're done in combination with, targets for PD-L1 or the CTLA-4, as well as some other chemotherapeutic agents at times. And you will notice also that they are all in early phases. These were selected as they are targeting solid tumors, but trials in immuno-oncolytic viruses actually are in numerous oncolytic viruses that have been identified using many different combination therapies, but by and large, we are still in our early phases of development in clinics. So while I'm in clinic doing or in the OR doing robotic surgery for stomach cancer patients, my lab, led by Dr. Fong, he's our PI in the lab, lead PI. There's a couple other PIs. We work together and study oncolytic viruses. So for the past few decades, even before I did my research in Fong Lab at Memorial, Dr. Fong and his team had been working to overcome some of the challenges to oncolytic therapy. There were lots of off targets in the beginning. There are physical barriers to tumor penetration of the virus. Antiviral immunity. Viruses are cleared by our system very quickly. And so we have to overcome that for oncolytic viruses to reach cancer. And because we need to balance the virulence with efficacy, a high dosing was required, which means that we have to produce a lot of virus to be able to treat one patient. And one of the other issues is that a lot of the oncolytic viruses in development, they took so long to get to clinical trials and be actually put into humans that they're running out of IP and no longer was being developed until very recently. So in the very recent years, our lab has developed a new virus that harnesses an immune modulation. And it's an interesting virus. The way it was created was that multiple viruses, orthopox viruses, are mixed together to generate 100 recombinant viral chimera. So these viruses actually, the recombinants do not naturally exist in nature. They were placed through high throughput testing of NCIS 60 human cancer cell lines, and we were able to identify 2 very potent oncolytic viruses, chimeras. One was CF33, the other little brown dot there was CF17, and we decided to take CF33 to clinic. So CF33 was selected for clinical translations. It has a unique double-stranded DNA viral genome that does not did not previously exist in nature. It was completely sequenced, and we found that the genetic mapping of this virus shows a mixture of orthopox virus groups that are well known and well known for their safety and not human trophic. So rabbit pox is rabbit tropic, and raccoon pox is raccoon tropic, and really should not be infecting humans, but it kills cancer. So they're fairly safe with limited tropism for normal human cells. And as you can see on the organ titration graphs on the bottom here, intratumoral treatment gets a lot of virus into the tumor, as well as in intravenous treatment. And in early phases, some of the other organs are actually infected. Over time, with intratumoral treatment, there is no more viral presence and gone, and usually measured in weeks. And it is very effective. Our studies have shown in vitro that it is very effective in treating triple negative breast cancer, pancreatic cancer, diffuse gastric cancer, lung cancer. So we have done preclinical studies in various solid tumors, including what I mentioned before in colorectal cancer. What we found was that CF33 backbone, the wild type, is very potent and is actually much more effective in killing cancer, both gastric and pancreatic, at 3 to 4 doses logs higher than the oncolytic viruses that are currently in clinical trials or in studies. Oncovex GFP, which is the backbone of one of the viruses, TVAC, and then the GLV1H68 is a vaccinia virus. So using low doses of PFQ pack forming units, which is how we dose oncolytic viruses, for immunocompromised animals, they're as low as 10 to the third, and immune competent model, we need a higher dose, but these are 2 to 3 logs lower than what is known for other viruses. We edited this virus CF33 for safety and for oncolytic virus tracking. So we took out I didn't do it, the virologist did it, the thymidine kinase, and then we were able to place a EGFP, fluke, HNIS. The first two for preclinical, all three for preclinical testing, but HNIS is FDA approved for use in humans, and they will be in the virus that is going to clinical trial. What we did was we tested to make sure that the virus transferred genes actually express proteins that are functional. So biominescence tracking of oncolytic viruses and tumor in vivo can be used with fluke expressing CF33, IV injection with oncolytic virus. CF33 fluke allows us to do bioluminescence imaging, tracking tumor as they grow and are infected by the virus. And as the treatment works, you can see that over time, the tumor and the virus disappear. Same thing in IT injections of one of the tumors that you're seeing in the PET imaging of CF33 HNIS infection. One IT injection of one of these tumors leads to the virus tracking to the other tumors. And you can see that over time, you could see the PET imaging change with the virus infection and killing of the cells. The thing that I'm working on that I will not present to you today in the interest of time is what we're doing with the oncolytic virus with peritoneal washings and malignant ascites to see ex vivo what is happening after virus infection. We can see that the virus CF33 GFP pictured here with near-infrared imaging, actually it shows that it's infecting something. Whether these are cancer cells or immune cells are still to be analyzed. So the thing with GFP is that with the in vitro studies, we can plate the cells and infect them at different MOIs, and over a period of time, you can see the virus expand in the cancer cells, replicate and express the transgene. and the virus disappears, the GFP expression disappears as the cancer cells are dying, and it corresponds to the cytotoxicity data of cell survival percentage over the course of time with different MOIs. Very useful for preclinical testing, but GFP will not be in humans. The studies that were done over the course, obviously, this is Michael O'Leary. He's a fellow that used to be in Fong Lab, who finished his clinical fellowship in surgical oncology with us, is now in a full-fledged attending. When he was a fellow in the Fong Lab, like I was in the past, he worked with the CF33 and found that you can inject one tumor and obviously tracks to the other. And this is what I was explaining to you before. These papers are published, not just in pancreas cancer, but in colon cancer and many other solid tumor types, cancer types. And what is, again, interesting is that the virus can track to the other tumor, but is fairly safe in the dosage and the biodistribution and multiple logs. So the safety and the efficacy and the ability of the virus to track to cancer has been confirmed over multiple preclinical testing. The most exciting thing that was found, actually, one of the most exciting was studies that were done led by Sham. Sham is our immunologist, the virologist in a lab, the man that creates our genetically engineered variants. He worked with triple negative breast cancer and in syngeneic mice and using mouse-specific anti-PD-L1 antibody after virus infection. He found that there is an upregulation of PD-L1 after virus infection. So CF33H-ness delta F 14.5 has 2 deletions and 1 insertion of H-ness gene, and the other one is blank. While it looks like there's a lot of upregulation, statistically, it was not significant. However, if you look at the co-inflammatory factors like interferon gamma, IL-6, and tumor TNF alpha, peritumoral environment, these are all upregulated, whereas in the serum, they are not. So there's a fairly local regional response that we see. And what Sham did was to treat the animals with multiple doses of CF33H-ness, and then plus or minus the anti-PD-L1 in combination and mouse anti-PD-L1. And what we saw was that the tumor volume is the least growth in the combination therapy, as well, corresponding to survival benefit. This is a very exciting finding to show that there's potential combination enhancement with virus and anti-PD-L1 checkpoint inhibitors in cancer therapy. So we checked the PD-L1 status in gastric cancer cell lines. Most of you who study gastric cancer already know that this is the case. We infected it with virus and learned that, interestingly, while the PD-L1 expression level may not overall increase, the cell surface expression or staining is increased after viral infection, perhaps indicating that intracellular PD-L1 is moving to be expressed on the surface. So Dr. Zhang, who is an immunologist in the lab that I work with very closely, we work together on looking at the effect of our virus in pancreatic cancer. Same effects are happening in vitro for these infected cancer cells. After virus infection, we're able to upregulate as the cancers definitely die, but the remaining cancer cells actually have upregulation of PD-L1 by day 5. And this effect is also true with interferon gamma. So what we'll notice here on the right is that this just staining for PD-L1, nice pictures of upregulation after virus treatment and interferon gamma. Why this is important is that these findings led us to rationalize the need for a potential oncolytic virus CF33 backbone that carries with it the gene expression cassette for anti-PD-L1 for local regional delivery, surmising perhaps if we provided the virus along with anti-PD-L1 in the region of the cancer, that they would be a combined efficacy. After Sham created this virus with the 2 deletions of the virus gene, we injected the 2 gene insertion, HNIS for imaging and potential radioiodine therapy, along with anti-PD-L1 cassette. We confirmed that infected cells, in fact, create this protein. Anti-DDDK tag binds the virus, produce single chain anti-PD-L1 protein. They're both existing in, so the anti-PD-L1 protein actually exists both in the lysate and a supernatant of the infected cancer cells, and they release it as they are killed. Functional assays are done to confirm that these are not just proteins, but they are functional proteins. We are currently working to move HNIS anti-PD-L1 into the treatment of GI peritoneal carcinomatosis, hoping that this would turn some hot immunosuppressed environment into a more immune hot environment. In vivo studies, first in immunocompromised models, were done, and ex vivo studies with washings from malignant ascites from gastric cancer patients are also being done. Briefly, we just confirmed that CF33 anti-PD-L1 is dose dependent and very effective in humans and not so effective in mouse models, which is one limitation of doing syngeneic mouse model studies. It is not always, the virus does not always infect and replicate, although it does for majority of the gastric cancer cell lines. You can see that there are some resistance, like SNU1, the virus infects, but it doesn't replicate. And it translates into less effective killing of the cells, although at higher doses, all of this disappears. And so the same kind of studies were done in vitro looking at anti-PD-L1 effects. And we know that the virus, once you upregulate with TNF gamma PD-L1, the virus, it can infect and release in the local area anti-PD-L1. And you can see in the yellow arrow that the virus released PD-L1 has bound to the cancer cell, and they co-localize. And there is a significant difference in the expression levels of PD-L1 with interferon gamma, but once you give the virus with anti-PD-L1, the PD-L1 levels go back down. So after confirming that, we sort of turned the immune therapy away for a second to just understand what happens to the virus in terms of route of delivery and oncolytic efficacy. What you see here is a published paper showing that with IP and IV treatment of the virus in immune compromised mouse with flank and subcutaneous and peritoneal tumors, that IP is more effective than IV in ensuring tumor regression and animal survival. As you can see here on the right, the IP animals treated on day 2 did much better than the IV or PBS treated animals. This and we actually repeated the study so that we can get some imaging and find out what's going on with the virus after IT injection. And we can inject the subcutaneous tumor, and this virus will track to the peritoneum and express both HNIS for PET imaging as well as anti-PD-L1, which I did not show here, but we have confirmation that it does. And that IT injection prolongs survival and can actually eliminate peritoneal carcinomatosis in 2 of the animals that were tested. So what are we doing in the next steps? Well, we want to identify immune biomarkers for this oncolytic virus. We plan to continue our work in syngeneic mouse models, although it may be very difficult as the higher doses are required. We're looking to acquire a transgenic genomically stable mouse model for gastric cancer to test the virus. And we are completing our genomic studies of with the CF33 virus ex vivo treated peritoneal cells. Where are we in terms of getting to man? As the virus was licensed out, I think Immugene is planning for clinical trial early next year. So intellectual property was done. Toxicity and safety is completed. Preclinical studies are being complete. The IRB IND is currently being written and submitted. The GMP for products, we actually have some and can be done on campus at City of Hope. Obviously for the ongoing studies done preclinically and some of the trials, other than Immugene funding whatever they do, we're looking for grants, and it's going to be a long process before FDA approval, and it becomes, after first in man, looking for what is next in clinical trials. So phase 1 trial planned in GI tumors in the failed setting. Where do we fit into how will OV therapy contribute to the emerging body of treatment options for solid tumors, including gastric cancer? Patients who I treat, we don't know yet, it's still pending. And how will tumor-specific genomic profiles direct OV therapy? That is also unknown, but it is being investigated. And what phase 2 studies should be done is also being considered. We need to determine best methods of OP therapy, delivery, HIPEC, PIPEC as surgeons. These are things to consider. How will we combine this therapy with regional or bidirectional therapy? How to monitor treatment efficacy, how to predict therapeutic response and outcome, and what is the next transgene to modify the virus, because a large viral genome backbone can carry a lot of things, molecular targets, immune targets, CAR T targets. And so what's next? We are partnering up with the CAR T team. Anthony Park is part of our group. And how long until the next novel effective OV gene therapy take to reach our GI cancer patients? That is unknown, but I'm hoping within the next year or so. In summary, after decades of inertia, the reemergence of OV therapy for solid tumors is being fueled by recent advancements in gene editing and molecular profiling to bring oncolytic viruses within immuno-oncology and precision medicine. CF33 platform of viruses are headed to clinical trial from our lab. CF33OVs demonstrate clinical promise and is known to treat treatment-resistant, genomically stable, and non-immunogenic tumors. IT route of delivery seems to be best for both primary and metastatic tumors in the peritoneum. We will next explore CF33 delivered cancer cell transgene expression of CD19 for combined therapy with CD19 CAR Ts. Emerging molecular profiling of peritoneal immune TME will really provide us with novel targets for additional combined therapies. I want to thank the Gastric Cancer Work Group for helping with our clinical bio specimen studies, and our research collaborators for teaming up to cure cancer, and the patient advocates and my patients and donors, as no life should be lost to stomach cancer or any cancer. Thank you for your attention, and please come visit Los Angeles when COVID-19 is over or under better control. I'm so sorry for going over time.
No worries, sir. Thank you very much, Dr. Wu, for that very nice talk again. It's actually very exciting to see the work that you are doing here because we know that this new age is the era of immunotherapy. And even though it seems very promising, we know that a large group of patients do not respond to current immunotherapy regimes, especially with regards to gastric cancer. So if your oncolytic virus therapy can help to improve and synergize with immunotherapy, it will be very promising and very helpful for our patients. I do have a question to you as well regarding your talk. You did mention that there's a difference in terms of outcomes with regards to the route of administration, meaning that when you give it intratumorally versus intravenously, there's a difference in the response and the outcomes. Have you tried the intraperitoneal administration as well? And what do you think is the mechanism behind the difference or the different responses with regards to the administration route?
Yeah, so in terms of the viral replication, because the virus feeds on being infected and replicating in tumor, the IT, I believe, provides the best like milieu for the virus to replicate and kill directly. So the IT seems to be obviously the most effective and probably the safest in terms of, its biodistribution. The IV is limited, and it's the host antiviral impact is not seen in the immunocompromised animals. So the virus actually gets to the tumor fairly efficiently and replicates and probably can travel around and find the other tumors. But in the immunocompetent mouse, and we know that in humans that it's cleared, the virus will be cleared. And even giving a second dose of virus, because of our immune memory, it may be cleared even faster. So we have to give a second dose fairly quickly. I mean, after within days, or the antivirus memory of our immunity will clear it. So what about IP? Yeah, we tried IP. So one of the benefits of IP is that you can give a lot of volume into the peritoneum, but because the tumors are usually small and it's not as big in terms of the virus replication. So it takes a little bit longer for the virus to track to the peritoneal cancers, but they do. And what happens is over time, it actually comes into the blood and can track to the primary tumor. What we've noticed is that in the immunocompromised mice, the IT is the most effective for even peritoneal carcinomatosis, for even over IP. IV is the worst. And IV is just not as good overall, but it is what we're used to. Chemotherapy, immunotherapy, all of this is given mostly IV. And so when I talk about designing clinical trials and I say, oh, let's give it IP, they say, oh no, that's not the first route that you can get FDA approval for. So it's going to be IT probably for the time being, and then possibly IV. But we're making every effort to try to get this, ultimately in phase 2 trials to be IP somehow after getting FDA approval for use. But IT first, probably.
Thank you. Are there any other questions for the panel?
Sorry, just one question for me. I guess, as you mentioned, IT would be the best route of giving it, but that can be quite challenging in peritoneal carcinomatosis because of the multiple sites of disease. Have you tried like a dual modality of giving it, like say IP and maybe IT?
Yeah, absolutely. I think there are two. If you consider a patient with their primary tumor intact, like pancreatic cancer with peritoneal metastases or gastroesophageal colon with peritoneal metastases, then we can get to the primary tumor and inject. Like there's a virus trial right now with endoscopic injection, not with our virus, but with a VSV. And so intratumoral endoscopic injection of, you can even do EUS for the pancreas, right? And so, but in the absence, if patients are recurring in the peritoneum, absolutely should consider IP over IV, I think. And there are many ways to give. I mean, we can do catheters, we can go in ourselves as surgeons and do it. And so, yeah, I would love to see very versatile treatment, would like a route as we go forward and find out more about how it is impacting the tumor microenvironment.
Okay. And I guess a lot of your data was shown in GI cancers, mostly gastric cancer. But what has your experience been with the ovarian cell lines?
Yeah, so great. So actually the immune competent models that are done are in lung and in triple negative breast cancer and colon, because they just exist much easier. And the ovarian has been studied. It has similar responses. And obviously the ovarian model is being, it's actually being done in conjunction with another lab because we don't have gynonc in our lab. So there's another lab that has been studying this with us, and it's similar. They try IP, obviously it's like the first for gynoncs, like it's like the first thing. And so it's efficacious in those doses that I showed, very similar. The mouse, unfortunately, when we have syngeneic models, the dosing is so much higher, you know, like 2 to 3 logs higher required to kill the like mouse cancer cells. So mouse ovarian cancer cells require a much higher dose, requiring a lot of virus to do, like if you have to do about 50 animals, it's like logs and logs of virus. And so we're being very cautious about, you know, how to use the virus in our animal models.
Okay, thank you.
We have a question from the floor. Professor Jimmy So.
Hi, Yanghee, great to see you online. And
hi, Professor So. Hi, Jimmy.
Happy to
Thanks
see
for the
your,
invitation.
Happy to see
yes,
your
I
progress.
wish I was there.
Sure, great. Yeah. Very basic questions. Most of us are surgeons and not virologists. How come your oncolytic virus are so smart? They can target the cancer cell, not the normal cell?
Yeah, so that's a great question. So I think for every oncolytic virus type, it's different. So there are some unknowns in oncolytic viral therapy, and these are one of them. But one of the things that we know about, for example, for the herpes oncolytic virus, is that there is a cell surface marker like nectin and others that it's altered in cancer. And so as the virus goes around, it actually likes to preferentially go into these abnormal cells because the cell surface is different. But what is distinct about the oncolytic viruses all around is not their cell surface or how they enter the cell, but the fact that without an abnormal genome, they can't replicate. So overexpression of ribonucleotide reductase, etc., is the way the oncolytic viruses seem to take over the cancer cells. How do they find the cancer cells? I don't know. I mean, it's by blood usually or fluid. So I don't know exactly how that's done. How does herpes find its way to our brain and our nerves? It's sort of the same way that it finds its way to the cancer cell, I think.
Any other questions from the floor? Maybe just one last question for me regarding safety as well, because like Prof, we don't handle viruses on a daily basis, and a lot of these viruses are very scary and very worrying to most of the laymen out there as well, especially in this pandemic era. You know, just wondering how will this affect the use of oncolytic viruses?
So that was one of the reasons oncolytic viruses went off the plate was gene therapy. There was some badness that happened in, I think, like 1989 or 1995 with gene therapy, right? So oncolytic viruses went off the plate. And the oncolytic viruses that are now, they've been around for so long, and now they've been in humans over and over again and shown efficacy. Initially, the virulence was a huge factor, herpes virus going to the brain and being latent, for example. So they took out the 2 neurovirulent genes. Vaccinia virus is safe in that it was used to make vaccines for smallpox. So there's a lot of the strains that are used in our virus. We selected the pox viruses for specific reasons, the strains, because they're already known to be safe and have been tested. But it's one of the FDA's, it's one of the most important factors in bringing oncolytic viruses to humans, because the safety, and you have to prove over and over again, but the safety of the viruses that are out there, the therapeutic window is fairly high, especially for CF33. That's why CF33 is so unique. It requires such a low dose, and it's fairly safe. But the large animal studies have to be done, and to get the FDA regulation approval, even for an IND, to bring this to humans in phase 1 trial, it's like thousands of pages of regulatory paperwork. So it is very carefully considered.
That's very reassuring to hear. Thank you very much, Dr. Wu.